Thank you for being here this morning. I really appreciate all of you. And now I'll turn it over to Jennifer Roggenbuck. Do you have a speaker mic?
I think I mic'd up already. Can everyone hear me?
Okay.
Okay, perfect. Met many of you yesterday, and I'm so honored to be speaking here along with Dr. Anna Sarkozy and Peter Hackman about our research in Titin disease. And I came to this area of study through clinical dilemmas that I encountered in the Muscular Dystrophy Association clinic where I work at The Ohio State University. And when I began at this clinic, it was about 10 years ago, and it coincided with the availability of next-gen sequencing for clinical genetic diagnostics. So I started in this clinic, and I was faced with a large backlog of patients who didn't have a diagnosis and had been followed in that clinic for many, many years. And my boss at that time was Dr. John Kissel, and he would always tell me, find a diagnosis or you're fired. And that was a joke, but still, it was a little bit of a stressful situation. But one of the individuals that I met in that clinic was this man here, and he'd been a patient of Dr. Kissel's for more than 20 years with a diagnosis of limb-girdle muscular dystrophy. And he'd had some testing. He'd been tested for FSHD and myotonic dystrophy and a few of the more common limb-girdle conditions, but didn't have an answer as to what was going on with his weakness. And I met with him and learned that he had recently been diagnosed with dilated cardiomyopathy. He was about 70 years old at that time. At that point had a 20-year history of weakness, but also had recently got dilated cardiomyopathy. And then when I took his family history, he told me that his son had something called tachycardia-induced cardiomyopathy, and that he was discharged from the Army because he couldn't do push-ups and he had a CK elevation. So that was all very interesting, and we know that the heart is a muscle too. When I ordered his genetic testing, he had a single variant in Titin, which is a variant that affects the length or the predicted length of the protein. We call it a truncating variant, and this kind of variant is well known to cause cardiac disease, as we heard yesterday in the cardiac session, but it had never been known to cause weakness in the skeletal muscle or the muscle of the body. So that is, kind of a mystery as to, what was this, was this the single cause of his weakness? But male-to-male transmission from father to son is a hallmark of autosomal dominant inheritance. So anytime we see that in genetics, we think, well, this has to be a dominant condition. And as we also heard yesterday, we've got two main patterns, or two or three or four main patterns of inheritance in genetics. And most limb girdles are dominant or recessive. And dominant means it can be passed down from generation to generation. Recessive means it takes that combination of a single gene from parents who are typically carriers and unaffected. So I'm going to skip this because we already went through this before. And when we have proteins made from genes, there is a little bit more detail that I'd like to tell you about. The top cartoon is DNA, and that shows the double-stranded DNA that forms the basis of our genetic code. Most of our genes are broken up into these coding units called exons. And then when the body goes to read that information that's encoded by the gene, it makes this temporary copy called RNA. And RNA is almost like your copy of the recipe card that you take through the kitchen, and you make the recipe based on that. And when an RNA is made, it's single-stranded. It leaves the nucleus to go out to the cytoplasm to make the protein, and then it gets chopped up and stitched back together in different ways. And this process is really important in Titin and results in the different versions of the protein or isoforms that exist in Titin. This RNA also provides us an opportunity for diagnostics as well as therapeutics. So that's why I wanted to say a little bit more about RNA. To go back to our diagnostic dilemma, there's only 3 known dominant titinopathies, or at least at that time, when I was seeing that family. On the skeletal muscle side, there's tibial muscular dystrophy, as discovered and described by Dr. Hackman here, and another one called hereditary myopathy with early respiratory failure. Now, this patient didn't fit with either of those. And on the cardiac side, of course, we have the very well-known Titin-related dilated cardiomyopathy. And according to the literature that I was reading as a novice trying to help these families, all remaining Titin-related skeletal myopathies are recessive. So it was a real dilemma. Like, we clearly had dominant inheritance in this family, but it didn't match up with any of these known dominant titinopathies. So we heard a little bit yesterday in the cardiac research session about what is the mechanism of disease in dilated cardiomyopathies. And in dominant conditions, there can really be 2. Either the protein that's made is not enough, that's called haploinsufficiency, or there's a bad protein that gets made that kind of messes with the works, and the cardiologist yesterday referred to this. So when we have recessive conditions, it's usually a little bit more straightforward. It's usually some kind of loss of function, meaning that the gene is not making enough of or functional protein. So when we have a dominant condition, we don't automatically know what the mechanism of disease is, and that's actually important for treatment. So to go back to the families that I was studying, I kept identifying these families where we had dominant transmission of a single variant with this cardioskeletal phenotype, meaning that they both had skeletal muscle weakness and often would develop cardiac symptoms later in life. So we tracked these variants through the family and also collected muscle tissue via a needle biopsy, so a non-invasive kind of needle biopsy, to do various studies of RNA, protein, and muscle fiber mechanics. And we teamed up with Dr. Hank Granzier's lab at the University of Arizona for some of that work. But here's a picture of some of the families that we found. And basically, we had single Titin variants that were, in many cases, truncating, but there were also some deletions, and also some splice site variants that segregated in families in an autosomal dominant pattern. And we did check for harmful variants in this SRPK3 gene that was newly identified and did not find those in these families either. And then as far as the RNA, we wanted to see, number one, like how much RNA is made in these cases, and then were there any splicing abnormalities or other differences in the RNA. And just to kind of, cut to the chase, we did see that in our truncating cases that the RNA levels were lower. So these individuals were not making as much RNA from their Titin genes. Now, we also saw some interesting things with the deletion case. There was one deletion case that had an end-frame deletion that spanned a crucial part of the A to M band, and there was dropout from the affected exons in that deletion case. So it matched up exactly with what the DNA said. So that was kind of a nice thing to see because it was kind of a control that's telling us that our RNA-seq is working correctly. However, to our surprise, the protein levels, the amounts of protein, and the apparent quality of the protein was the same in these truncating variant cases. So this is a paradox. If the RNA level is lower, shouldn't the protein also be lower? That's not what we saw. So somehow the body is compensating and maybe using that normal RNA that's made from the other gene to upregulate that somehow and kind of crank up translation or the production of protein. So I'd love to hear from Dr. Hackman later about what the mechanism might be here, but apparently we see this in other muscle genes too. There was only one case where we did actually see a truncated protein, and that was that case with the deletion and the end-frame deletion. So that is an example of the classic dominant negative or toxic gain of function type of effect. But that was the only case where we saw that, and that therefore really the only case that we can truly prove a dominant mechanism of action in this cohort. So just to quickly wrap up, as far as the clinical impact of this work, We do believe that we've identified a new dominant titinopathy that's characterized by mild weakness of the limb girdle pattern. It can also involve axial or neck weakness. And some people have proximal and distal weakness. And then these individuals often develop cardiomyopathy or arrhythmias, especially as they get older. And this occurs in some single titin variants, but apparently not all. And interestingly and importantly, we can see mild dominant phenotypes and severe recessive phenotypes in the same family. So a child with a severe, biallelic type of titinopathy can have parents who might have just the cardiomyopathy or some mild skeletal muscle weakness as well. And there's now been several other reports as well. That cardiology study that I mentioned yesterday, where they looked at the skeletal muscle of truncating variant carriers and found increased fat fraction in their skeletal muscle. So conclusions, some pathogenic mechanisms of muscular dystrophy in the monoallelic state, or the single variant state. Even though the phenotypes of different mutation types seem to be the same, the mechanism of disease underlying, for example, the deletion and the truncating variants might be different. And we know from the heart literature and the heart studies that truncating variants are known to produce small amounts of truncated protein in the heart, but we did not identify that in our families despite looking pretty hard. We really tried to see if we could find any evidence for truncated proteins in the skeletal muscle and did not find them. So this the hypothesis is that this dropout of the RNA that we're seeing in the truncating variant cases, we call that nonsense-mediated decay, could have a protective effect because it prevents the formation of these toxic truncated proteins. But we are still looking into that, and we want to find out is if the RNA dropout or the nonsense-mediated decay might correlate with disease severity in persons with single truncating variants in Titin. And we hope to investigate this and other genotype-phenotype correlations in the dominant cohort that I have collected, as well as a cardiomyopathy-only cohort and then normal controls, because we do have some normal people who have Titin variants as well. So if we identify there's a protective effect from this RNA dropout or nonsense-mediated decay, that abnormal Titin RNA transcript could be a therapeutic target to prevent the formation of toxic protein, which is damaging to muscle function. So that is a very quick summary, and I hope there'll be time at the end to talk and ask more questions.
Thank you very much. Thank you, Jen. I really want to just pause and say a reflection. Thank you for that presentation. Is that you can pull up the next thank you, while they're getting ready. Is that I am just so moved, and you're going to just hear it in my voice that this is against the headwinds of what's thought to be true about titinopathy. And this is an example of caring about patients, people that you met, curiosity, and persistence to discover new findings about our conditions for our families for the future. And did at times face some pretty strong headwinds because this is not what's commonly held to be true about titinopathies, if you get what I'm saying. And so I so appreciate your work. And it's the same theme of Peter Hackman hand sequencing Titin back in 2000 using the old school reagents, and he didn't stop, and he kept at it. And the curiosity and the persistence and facing these headwinds. And one of our roles at Team Titin is to support research. And I'm just incredibly, and Anna putting together, you're going to hear about this natural history study to focus on the recessive titinopathies. And by the way, did take years for us to get this ball rolling. And so this theme, I feel you guys right here in my heart. And on our logo, there's a giant heart in the middle. And I extend that to you and thank you for your persistence, your curiosity, and your work. And I hope that you do. And it is one of our pillars of our mission is to support research. And I just really hope that all of you in this room can feel this, not just coming alive off the slides and hearing the words of our researchers today. So thank you.
Shall I?
Yes.
Okay.
And I'll turn it over to Anna.
Thank you. Thank you very much. Okay, so I'm going to present, give you a little bit of an update, an overview about this study that Sarah has just mentioned, which is a natural history study in particular for recessive Titin myopathy. Just we'll do a very quick background, just to remind you about that. We know that there are many different forms of myopathy associated with Titin, and we are going to focus really on the recessive forms. Does this work? No, it's all right. Doesn't matter. I will. The one? Turn it on. Oh, all right. Yay. So we heard yesterday about all the forms of titinopathy, dominant, recessive, and we just heard about the dominant form. But at the moment, this talk will be focused mostly on the early onset congenital myopathies that I was also reporting yesterday, that are recessive, that often present in early life, and that have specific characteristics with cardiac, respiratory, and feeding difficulties, and some specific changes on the biopsy. And I showed you yesterday this picture, and I wanted to, I bring this back because I wanted to highlight something. So clearly, what we aim for is to find a cure for titinopathies. But it's also important in the meantime to, in absence of cure, to trying to find the best management and treatments to help people with titinopathy. What is also important is to try to understand, in order to make changes, you have to understand what would be natural history of a condition without therapies, and then what is the natural history with the therapies that are available today. Because sometimes when you apply therapy, you could actually see already some changes. So for example, if you have cardiac medications or cardiac prevention early on, you might maybe delay onset of certain symptoms. And then of course, that can change what you would actually see in terms of natural history of the disease. And this is something that we already see in other conditions that have been studied for longer time. And again, knowledge from other conditions can help you to develop, interventions for less known conditions, like for example, titinopathy. So you really already will see that with some intervention, the natural history of titinopathy could perhaps be modified. Clearly, what we aim for is to arrive to this point, that you cure it. So this is really to highlight that we need to understand and clarify well the natural history with and without the available symptomatic therapies before we can really to help us to understand how to develop therapies. Another important aspect that I would like to highlight is what do we need really to become trial ready. So we need therapies, but what you need is to develop the patients who have titinopathies need to be trial ready, so need to be available, and you have to have some characteristic of the population, also of our knowledge, to make us ready to have clinical trials. So there are different steps that this is valid for any conditions, rare conditions. So really, first step is to understand natural history, and this is what we are going to talk about. But there are other aspects like optimizing care and having international standards of care to become trial ready. Again, it's important because if I give a medication or a treatment for people with a condition that are not receiving already standards of care, and therefore might be less healthy compared to, for example, a group of patients who receive regular medication, clearly the baseline where you start from would be different. So it's important that everybody gets the right therapies that are available today before thinking about even getting, therapies that can cure the condition. So that is very important. And then you have to identify the specific outcomes, measurements, like tools to measure change over time, and markers to say that a drug actually works. And when we talk about outcome measures, I'll give you some examples. So you may have physiotherapy tools, so assessment that somebody does in clinic when you come to clinic, like how long a person can walk, or what is the strength of their arm muscles, or how much they can blow in a tube, and you measure this, and then you measure it again, and you measure it again. And then you compare over time if these measurements change. So all of this can tell you what is the change over time, and if you use a drug, then you can compare the measurements with the drug, without the drug, and then you can tell you if the drug makes a difference. And you really have to understand how much time do you need to capture this change over time. So for example, if your ability to walk 10 meters doesn't change in 6 months, but it changes in 12 months, if I have a drug and I want to test the benefit of the drug, if I only study it for 6 months, you will not see a difference because that person wouldn't change in 6 months. But if I measure the drug over 12 months, I might see a difference compared to who does not take a drug. So you really have to understand how long do I have to check the benefit of a drug. And then you can have other biomarkers that really show you the benefit of, for example, a drug on the muscle. Like, for example, I can do imaging, like we saw yesterday with MRI. In some studies, they also do biopsies, but clearly you try to find markers that are not invasive, where the person wouldn't need to have tests like a biopsy or anesthetics and so on. So really, these are all things that can help you to understand, to make a population trial ready. So to know what's the natural history, you want to identify the right outcome measures, and then most importantly, you need to have also idea of how many patients there are with a certain condition. Knowing the prevalence and the numbers of people with a condition, it's key, because clearly also pharmaceutical companies want to, when they think about investing in a drug, you want to show the numbers. And Titin seems to be much more common than what we thought. And having the right numbers, so you being in a community where people understand the numbers of patients with a condition can really help us to understand where, what is the power of in developing this drug. So it's important. So what we try to understand and to help with to become trial ready is to really understand, first of all, the natural history and the outcome measures. So this is where our study has been focusing. And in our center, we had quite a lot of interest in understanding natural history of condition, in particular of rare myopathies and muscular dystrophies. And we developed this natural history study a few years back with support from Team Titin and from the German Association for Central Nuclear Myopathies. And the study has been designed in our department in Great Ormond Street Hospital together with Giovanni Baranello, who is one of the consultants in Great Ormond Street. Amy Wolf is our physiotherapist, and Professor Montoni is the head of the department, and Tishal is our study coordinator. And there's a second team in London that is involved, is led by Professor Jungbluth, and with Jenny Sheehan and Maria Vanegas, who are the other two collaborators, physiotherapists and clinician working in the team. The study also involves an imaging team, radiologists who will be involved into the understanding of the MRI aspects. What are the aims of this study? We want to understand better the natural history. So what is the course of the disease over time? And understand what are the best tools, these markers, that can tell us how the condition changes over time. And exactly understanding these little changes in 6, 12, 18 months to tell us if this can help us to understand the effects of drugs. And then we also wanted to validate imaging like ultrasound and MRI, that in order to understand if this investigation can not just support diagnosis, because we heard yesterday the diagnosis can be complicated sometimes, but also if MRI and imaging can track change over time. So if I look at an MRI at 0 at 12 months, do I see any difference at all or not? So for this, we developed a protocol, which this is a summary of what we do. People will come to clinic to have medical assessments, respiratory assessments, physiotherapy, and imaging. And this will be done every 6 months. And we originally started with at least 18-month study, but now we had further funds and we are able to go up to 2 years, and in a few patients up to 2.5 years. And we aim to arrange all this assessment in clinic and then track it in a very detailed way. And then we record all the assessments, and then we will analyze all the data at the end. The medical assessment would be a clinical neurological assessment where we look at all the clinical signs and symptoms. Then patients would have respiratory assessment when they are old enough to do it. These are all children, because we're in a pediatric hospital, so sorry, I forgot to say that, but these are all for children because we unfortunately cannot see adults in our center, but we will see everybody from 0 to 18. And if they are aged above 5, we will do respiratory assessment that are a little bit more in detail than what we would usually do in clinic, because we also want to understand if some of the more in-depth respiratory assessment could be better and more useful for trials. And then a range of physiotherapy assessment, again, to understand which one of these is the best outcome to measure change over time. And then we will do ultrasound and MRI wherever is possible for the child without needing anesthetic or anything, so nothing invasive. So where we are at the moment, we have recruited 21 patients, which was really good, and really I'm so grateful for all the families who have accepted to come to the study, because they do it, as you can understand, this is a gift that they give to us to learn more about the condition, and they're all under age 18. So far, we believe that the measurements that we chose are feasible, and we didn't see that any of the assessment was too cumbersome, and that we were able to keep majority of the patient in the study. There was a family who was coming from north of Scotland who decided that it was a bit too far, not because of the test, but it's just the distance in the UK was a bit too much. But otherwise, all the other patients are continuing the study. We are doing all the physio assessment that I mentioned. We are starting to analyze the data. Just to tell you, there's already some results that is interesting. We always thought that fatigue was important, and we believe that fatigue is something that people often report, but fatigue does not seem to affect our outcome measure. So we cannot record fatigue when we measure it in the assessments. It's important for us because that's already something that we have been able to see at the baseline. And then we also started to do the respiratory assessment. We analyzed the baseline data. Again, very interesting data on how much is the respiratory function at baseline of these children. And now we are going to analyze at 6 and 12 months and so on. And ultrasound so far and MRI was very helpful because in majority of people, we actually see the changes in the MRI that typically are observed. So again, that can help us to support the diagnosis in all our patients. There are some challenges. So these are rare diseases. So we really had to identify every patient possible and ask, would you like to be part of the study? And as I said, they have been very positive and majority of the families were helpful. But as you understand, if you want to have a large numbers for natural history studies, it's difficult. So we could find 21 patients in the UK, but clearly you might need to do these studies on a wider cohort of people and countries to have strength in your study and the results. There is a lot of variability. If you have 21 patients, you might see that each of them have different symptoms and signs because the condition is like this. But nevertheless, the results are always useful. And sometimes we might see at the end that perhaps if you only have studied a condition for 6, 12, 18 months, you might not see changes. So, but even this result is a result. So it's something more than what we knew before. And so again, there are some solutions. We really are looking into extending our study to either side to have more information. And we also try to, in the meantime, we are doing a lot of genetic testing to identify more patients that might have different symptoms and maybe that we missed so far. So improve our diagnostic abilities. And in the meantime, collect also information from routine clinics, also for patients who are not in the natural history study. So we are continuing the study. We are now at almost a year and a half mark, and we will collect all the long-term data and compare the baseline to the 6 and the following months. And we are here to share the results with the community. We are preparing papers to get it out, so to show our data, because that's the aim of all of this, so that we can use this data to also help in care for patients, but also to share this information to make trial readiness a bit further and closer to reality. And yes, so we are also open for, if anybody wants to know about the study, please contact us. But we're also open to other clinicians and other teams around the world to share our protocol. And if anybody wants to replicate, that would be great. Thank you.
Thank you, Anna, very much. And so as we get ready for the next presentation, I just wanted to comment. Thank you very much on that. Come this way. Thank you very much, Anna, for your presentation. And just wanted to give some resources related to some of the things that she talked about. One of the things Anna was talking about is kind of leveling the playing field for our families with care. Because if you go in for a treatment and let's say you had an undiagnosed nighttime hypoventilation, which is a breathing disorder at night, and then you get into a clinical trial and the team says, oh, we're going to provide this treatment for you, but by the way, you actually need some nighttime breathing support, and they implement that. Now you've implemented 2 changes. And so, for the scientific method, we kind of have to understand what changes are you making to have an effect on your health. And so we want to level the playing field, first of all, because you probably feel better and live your best life getting the best care. And so during this SciFam, we did present many sessions on optimizing care, which were in the plenary sessions yesterday. And the other thing is we talked about resources for care and care guideline information on the Titin website, and we have a whole page on care, and includes cardiac care resources, and there's a brand new limb girdle care guideline, and then also the care guide for the congenital myopathies. And then Cure CMD has a guide for the congenital muscular dystrophies, which is a somewhat different subtype, but it's also packed full of great information. So we really want you to be living your best life, getting the best care for today, for many reasons, including as part of research and our pursuit of a treatment ultimately. And that is also so valuable for the natural history study, and identifying these outcome measures or tests that we need to kind of prove an effect, like how are you today, you get a treatment, how are you tomorrow? And so we really appreciate all the work that went into it. And so I'm going to turn it over to Dr. Hackman.
Thank you very much and good morning. They are nice to see that at least so many of you today. Yeah. So anyway, yes, some research updates, and there's some slides on the next one, I think, because these we went. And so it's about recognizing the pathogenic Titin variants, and we talked about this already yesterday. That I mean that the Titin-related skeletal muscle types, they set muscular, as you know, and you have experienced, and there was not that many dominant titinopathies that was related to skeletal muscular disorders. So there was the TMD that we have discussed, the Tibial Muscular Dystrophy. Maybe we can mention one thing about that we thought originally that it's really a Finnish, that it has arisen in Finland. But actually now we have had some collaboration with an Estonian group, and we have seen that it's also prevalent in Estonia. And then we made some more research. We did linkage research where you can see how it's linked to what area in the genome and so on, and how long the area is. And it seems like the area, I mean the positional area is smaller in Estonia, a little bit smaller in Estonia. We are still confirming it, but that would mean that it has actually arisen in Estonia and then came with people to Finland. So we call it now Finn Major, but and we are not going to change the name, but it should maybe be called Estonian Major or something like that. But that's another thing, and I plan to have a poster about this, but then we are a little bit still working on that, so I didn't, but I can mention it to you here, so it's not any deal. And then we have the H-Murph, which is partly dominant and so on, but and then we have seen truncations in Titin, but still a couple of years ago, maybe a few years ago, we thought that truncations in Titin will not cause any skeletal, or is not causing alone any skeletal muscular dystrophy, and I'm going into it more closely. But we found actually in some of the cases, we found some small subtle missense mutation, small mutation in the other allele, and then we could see that, okay, this might be the cause of the disorder, so that it's not really dominant in this case, but it's just that we overlooked the small mutations on the other allele. And the reason why we thought that it is that we thought that it's like this is that it happens this sort of nonsense-mediated decay, as you talked about, and that means that that when the RNA is produced, if there's something, if a mutation is causing a truncated, shorter RNA, then it will be destroyed in the cell before it expresses. And so we thought that that doesn't happen if it's at the end of close to the end of the gene. So then it will still recognize it as an RNA, and it will not be destroyed. So we thought that at least truncating mutations that occur earlier probably would not cause anything. But then we realized one thing, and that was that this nonsense-mediated decay is not complete. So that even if you have nonsense-mediated decay, which we have seen, we could still see on the RNA level that you have some extent of the other allele. So it doesn't happen completely. So there I don't know the percentage, but can be 10-20% there can be of this other allele. So that means that it is actually expressed even in the cases where you have it very early in the gene, but it's just to a lower level. So this is not like it's not completely like this. That's one thing we realized, and then we actually let's see if I had this slides there about that we actually, I don't have it written here, but then a few years ago we actually saw a family where we had a truncating mutation on one allele, nothing on the other, and it actually had muscular dystrophy. So it can also, it doesn't have to necessarily be together with a missense. Often you should first look at the missense mutation in the other, but it can happen also just by the fact that it's truncating and it's expressing to some level the RNA, and then it makes a protein that is not working. So it's not like so we thought first that this is not the case ever, but then we realized that we have to modify this a little bit. But if you see this co-segregating monoallelic Titin mutation, first of all you should check in the family that at least in the skeletal muscular dystrophies, it will always affect the person sooner or later. So you can see by segregation studies that that's everybody in the family who actually has this mutation, truncating mutation in one allele and nothing in the other. Do they actually all of them have the disease sooner or later in their life? Or and it's also that there are not any cases where they have something, of course, and they don't have this. So you should see that it's actually going together with the disorder. That's important. But sometimes this can be a little bit tricky because it's very late onset. Some can be very late onset, so you might not see anything until the late Middle Ages or something like that. And so this is why it's not so easy to always detect it. But then it's good to be sure about this. And then you have to, that it's really just because of this mutation. And then you can do these RNA level studies and see that to what extent is this truncated form expressed on the RNA level. And if it's expressed more or less on the RNA level so that you can detect it, then you know that, okay, it's possible that it makes a protein and actually causes this dominantly. And in that case, it's even better if you can also do it on the protein level, these studies. So you can do this so-called Western blots or some other studies, and there you can see that if you see it there as a protein shorter band, then you can be sure that yes, this is now the fact that it's causing it dominantly. But you, in my opinion, at least for the skeletal muscular dystrophies, you should have these supportive studies and so I mean these supportive results before you can for sure say that it's actually the cause. So I mean that's just good to keep in mind, and this goes for the skeletal muscular disorders. In cardiac, we are not dealing with the cardiac in our group. So I mean, and I know that many of these have been reported, but it's just good to keep in mind that you should check it on the different levels, and if it holds, yes, then it is. The other thing that you were talking about is this diagenic disorders. And sorry, I will just. So diagenic disorders where you have titinus, variant titinus 1, and then you have some other gene variant in some other gene as the second cause, so they together cause the disease. So they have not been found so much earlier, and that was just because of the methods that I mean, before there was good I mean, new next generation sequencing, it was really difficult to do this. But now when you get all the results from the whole genome, it's more easy to find these sort of things. And also people were not looking at it. They were happy when they found something that is related, and that's fine, and this is the cause. But now, because there are many cases that were not solved, and that means that you have they pop up more easily because you have solved all the easy cases that were dependent on one variant in one gene. And the difficult cases, part of them are diagenic. And now that is why they are now popping up, this sort of diagenic cases. So it was mentioned this SRPK3, and there is also one other that is called TRAPC11, for instance, and be more. Years it has been popping up, this sort of diagenic. Being solved. Let's see there. Yeah. But anyway, so also you can look at the frequencies of this variant, and that gives a hint if it's made. If you have a dominant variant, you should check that its frequency is low enough in all databases. It should be like 10 up to -7 or less. Then you can think that it's a dominant disease. And why is it's good to if there are too many of these, or it's too frequent. And but in the recessive case, when you need both alleles the disorder. So when you're screening for for these relevant variants, you look at the frequencies of how often they occur. So if they occur seldom enough, if they are rare enough, then they might be the reason. Because when you look through these next generation sequencing results, you see loads of variants. And then when you filter it according to these, then you get a much smaller list that you can really look at. And then computational tools available now, which can also detect sort of which were a bit tricky to detect before, and I'm not going into the theory of that, but it's more easy to detect like small mutation than these copy number variants. But now that has also become more easy, and also one thing that is the development of the methods, this single strand sequencing. Sorry, yes. So I mean, sorry, not single strand, but long read sequencing. So you get very long reads, and then you can actually see if there is something strange like a duplication or small deletion or something. And you can also get more reliable, you can distinguish if they are on the same strand or on different strands, the mutations. And that is also very, very important for the diagnostic nowadays. So this long read sequencing, so that's not the first thing you do, but if you don't find something, you can do that also. And then there's many of these search engines that we use, Varsom and so on, and then these predictional programs that I also mentioned yesterday, like alpha missense, for instance. And so using all of these methods mentioned, it's the reliability of the diagnostics is rapidly increasing, and that's why I said that time you can diagnose most or almost all of the titinopathies and also other. Is that we have, because of Bjarne Wood, he collected a lot of samples, not only from titinopathies, but from many other disorders as well. And we have continued to collect samples from all over the world. And that means that we have about 4,000 samples in our freezers. I mean, many of them are from the same families and so on, but we have hundreds of families. And that means that we have a lot of material still to go through, and that means that we will and continue to get more samples, but we will be able to find the cause of diseases in many more of them. So we have, like internally already, a lot of very good material to work on, and we can work for many years on that material. So that is, and it's done. I mean, we have a big group of these three. We had three muscular groups that were like fusion. I mean, not all of them are researchers, but also technical persons and other, and some of them are part-time and students and so on, but still a lot of people working on this, on different aspects, both from bioinformatics, pure genetic studies, and also these functional studies to use different models like cell lines. We also use zebrafish, for instance, that can be used not for titinopathy so well, but for some other disorders. And analysis should be done on the RNA level, on the protein level, to really determine if neuropathies will soon be possible, I think. Of course, you can have some difficult cases, but most of them at least. And the therapeutic aids are coming, but I know that's something that can be discussed. I mean, how many years it takes, and I cannot give any estimates, but it's not like science fiction anymore.
Thank you, Peter. I'm going to ask if all the panelists can come up to the front, and I just wanted a couple of things. One thing I wanted to say is we are at the tip of the iceberg of understanding titinopathy, and myself and my family, we're on this journey too, and I want answers, and I want better understanding for Titin, and of course, we want a treatment for the future. And what I want to say is that our community is more about finding the answers together than just getting the answers at this point. And so if you can hear from all of these speakers today that there were people who gave their genetic variants to the team in Helsinki. They gave tissue samples. There were families who, with young children, participated in a couple of hours of extra assessments at their clinic visits, and flew to Ohio and gave a piece of their leg muscle to Jennifer Roggenbuck, and those kind of things. So we as a community collectively are working together to find more answers. And so I appreciate all of you, all the researchers, for their work, and then also to the community that this is something we really need to go through together. So thank you. And then I also wanted to just mention about the abstract for the grant that it was our first request for proposals. Grant funding from Team Titin was through Isabel Richard and her team and at Genathon. And the poster from the abstract, it says, in this study, we use an innovative and powerful method to help characterize the outcome of specific mutations in Titin. It is based on tiny lab-grown muscles called organoids. And this is not science fiction, this is happening. And these were from real patient cells. And these muscles in a dish allow for studying the mechanisms of disease using the patient's genetic background to ultimately lead to personalized therapy. So you can read more about that, and I just wanted to draw that to your attention. So I would like to turn it over to you and more open discussion. Yes.
In our groups, but in our institute, some people are working on other disorders and using this, trying to do this organoids. But there are some difficulties there also. It's not so easy always. But, yeah.
Can you move in closer,
Yeah.
just so you're in the picture
Yeah.
thank you. Okay, so I want to turn it over to you. What questions or comments do you have from the community, or if you guys have more?
Thanks, Sarah. Hi, good morning. Thank you all again for being here. My question is I have so many questions, but I'm going to limit it. Folks keep talking about numbers, the importance of knowing the numbers. Gigi, who some of you may have met, who's now sleeping, but was here yesterday, is 18. So at the time when Sarah was setting things up, when I first met Sarah, there was like, I don't know, a handful of people that were talking about this. So I don't actually know the numbers. Like Gigi tells, she's like, how many, can you give me ballpark? Like, how rare is this? Because we also work with the National Organization of Rare Diseases, and there's a lot of confusion about what it means to be a rare disease. So when you talk about Titin, and furthermore, it would be good if you could describe not only the numbers overall that we might know, but the numbers in skeletal, cardio, and then both.
maybe I can start, and then probably somebody, my colleagues have also to answer, but giving numbers is often challenging because it depends on how you look at the numbers. And each of these studies to look at numbers is actually quite complicated. Complex in general, you need to do epidemiological studies and so on. The other complexity with Titin, that as you understand, many people may have a suspicion of a titinopathy, but you have one of these VUSs. So do you count everybody who has the variant in Titin, only those who have a pathogenic variant in Titin, or only those who have all of those who have a clinical suspicion of titinopathy? So you have to define your level. So behind you, there is a poster on that screen, which is actually from our group. And we looked at numbers of all congenital myopathy. We looked at people who have an early center looks at those. And 10 years is probably the second most common non-significant. If I would, the most common, but we believe that it's the rare. But if you add also the skeletal and cardiac and late onset, the numbers would become higher. So a rare disease has a frequency of less than 1 out of 2,000, so still rare.
And if I could just add, the only thing about this as well is that this is a scientific and obviously family issue, but it's also an advocacy issue,
Of
which
course.
Sarah's done an amazing job of this too. But for folks to understand how big the community is and what it is,
Of course.
even for practitioners, for them to understand Titin, they don't know what it is.
Just a final comment that I want to add, that unfortunately all of these aspects request a lot of funds as well. So for example, doing registries and curate the registry, there is so much effort to doing patient registry so that you can register yourself and say, I'm here. That takes so much, there is a lot of, so all of these steps unfortunately are cumbersome. And it's not that we don't want to do them. I wish we could have funds for all of this.
We have some efforts that I can talk about. It's an excellent question, and I know it's highly relevant, and so it's on my radar as Team Titin. And I can say we do have a registry, the Congenital Muscle Disease International Registry, and that is a way to sign yourself up and say, I'm here, and you can fill out the questionnaire so that we can, number one, count you, and number two, characterize you. What does your life look like living with Titin-related muscle or heart disease? And the other thing is, it is in a way that protects your privacy in how you give consent to share your own personal information. That's one thing. But what you're kind of pointing to is something both prevalence and incidence. And one of the efforts that we have been actually working on for a couple of years is working on something called an ICD code, which is a diagnostic code that is attached when you go to see a practitioner, and you get a code for a medical diagnosis. And it's been on our radar that it is one way that people count how many people have this given disorder in a particular region. The problem is Titin doesn't have a code. So we have had, you can pursue a code to the ICD coordination committee, and we've been working on that effort from 2019, meeting with different regulators, and we went forward with a couple of other organizations who got their code and we did not. And so we actually applied for a grant and received it, and we were in the process of moving forward with some experts, because it turns out that there are actually ICD code experts. And we have been waiting for, last summer there was an ENMC, which is a meeting of people talking about nomenclature, which is about how do you name the myopathies. And so we were kind of waiting till they have a consensus agreement, which consensus is one of the criteria to move forward to the committee, is to have a really well-defined plan. And so we're waiting for the publication to come out of that to move forward with some experts to pursue an ICD code. So when you go to the doctor, you can get that number. And other communities, like one of our groups, the Nemlin Myopathy group, has been successful. They were successful with some of the limb girdles, which Titin has a limb girdle phenotype. They only received an ICD code for some of them, and then everything else got put into a bucket of limb girdle muscular dystrophy unspecified. So we're kind of unspecified. But so that is one of the tools that we're working towards. The other thing is we have had some meetings with some experts at the Broad Institute that we're working with some of our other colleagues at this meeting to try to get some data using their tool that they have that's called Genie. And I had some collaborative meetings from the team in Helsinki with Marco Savarisi, along with the people at the Broad Institute. But there have been some barriers with the change in the administration. Is a couple of things. Is the ICD code planning committee was ceased operations for a while, and they canceled meetings, and they were not. So that was kind of a barrier for moving forward in that realm. And then at the Broad Institute, there were some layoffs, which resulted in delays because there's a lot of changes going on in the research environment in our country at this time. So we're really doing our best, but it is an effort that is on our radar, and it is not easy, like Anna said, because how do you begin to understand how many people with rare disease there are, especially when you can't even count them, because you go to the doctor and the doctor can't even put in your code. So the wheels are in motion. It's definitely on our radar, and it is important. And Anna talked about something called clinical trial readiness, and that is one of our efforts, is like, how do we look attractive to pharmaceutical companies that might have a treatment, and how do we as a community look cohesive and strong and have resources and tissue and animal models and have like a strong community and have some numbers and a registry, and so that you could look at retrospective data or all those things. Or how do you understand what measures are, how do you understand what the disease looks like, the condition looks like without treatment, so that when you introduce the treatment, you could prove that it works, and that is a requirement, you know? So we're like really working to make headway on all these areas, and I really appreciate every person who engages and participates, and the researchers, we thank the researchers, of course, who are doing the work, but it's really, we're gonna need to do it together.
So just one comment about this, how you define rare diseases. So it depends also on what area you are looking. So like Finland is an isolate, has been, is not anymore really, but has been an isolate, and that means that for instance this TMD, which basically is rare everywhere else in the world, is quite common in Finland. So it was like we have like 1 per 5,000 approximately. So it's not that rare in Finland, because you have like more than 1,000 cases there. But in the worldwide, it's very rare. And the same goes for different communities, like in the Amish community here, you have some diseases that are much more usual there than, so the countries are very different in this respect, and that's good to keep in mind. But then also that US is a big country, so you have all in all, you may have a lot of cases. It's just to sort of find them and get them to come join this community and so on, and then you have more impact in that way.
Just also wanted to add a comment about, in general, comment that although maybe you know, it's true, for example, that our studies on early onset myopathies, and maybe you see that other studies or maybe adult and so on, all this information at the end will be applicable and may have a relevance also for other presentation of titinopathy, because it's very likely that if there is a therapy, it will be more depending on what type of change you have or what is the function of how the drug works on the muscle. It may not be just for people who have a recess or you know, that is not, maybe the clinical presentation will not be the criteria to decide if the drug can work or not for that patient. So in a way, it would be great if we had every possible studies on all presentations, but every information is useful because it's one little step forward. Yeah, I just wanted to comment, make that comment.
So I have a question regarding if finding individuals with this Titin already diagnosed is a very limited resource right now. What kind of effort can the hospitals do? Like we go to Children's Hospital of Philadelphia. We go to a department which is catering to all neuromuscular diseases. So they do, and one of our case is just one of them. So is there a way to link up with all these different hospital systems around US at least to get this data pulled into your database? Is that an effort which is ongoing, or is that something we should probably look into?
Yeah, that's a great idea. It's a great question. The problem is that, of course, how would we do that?
It's help from the clinicians who are working on our case, right? If my clinician or the person who diagnosed me is willing to put in the data into your database at the time of diagnosis, with our consent, of course, because I know it's 2 different countries and 2 different rules and all that. But without the ISO-ID code being in place, can we do that? Like, is that something we can?
So the thing is that clinicians entering data into registries has been attempted in some groups, and it's limited. Now you can maybe do more easily with the electronic medical record, but with consent and identifiable data has to be removed. And so it's a huge challenge. And even Dr. Savarisi in Finland we had, going back since our ENMC workshop in 2016, made an international effort to expand a database. And it was extremely problematic because sharing data is, especially across countries, is extremely problematic because of privacy laws and protecting families and patients. So, you know, it can come from, for example, the free genetic testing program that's going through the Genetic Cardiomyopathy Awareness Consortium with every gene. As part of participating in that program, you consent to share all of your electronic medical records. That's a huge data source, and they de-identify it. And that's a huge benefit for working towards treatments. But it is very expensive, and it's very time-consuming because how do you de-identify an entire electronic medical record? It's a gigantic effort. And so it's being done in that cardiomyopathy space, and we might end up, because changes in the Titin gene are the most common cause of inherited cardiomyopathy, and unlike rare muscle disease, cardiomyopathy is quite common, that we might end up finding more progress in that field. And we, as far as looking attractive to industry partners who might be seeking a treatment, we might end up finding a treatment and through that avenue first because it's more attractive. And so that is one approach that it's been taken. And I'd love to collect the data, but how do I protect you and what your preferences for your family's data, and how do you collect that, and how do you store that, and who's organizing it? And asking, if you ask any clinician, like Anna's a clinician, Dr. Sarkozy sees patients in clinic, well, how much time would you have to be entering their data into a database? Now, we have some value and benefit for some countries that have a national health service because they have, you could probably speak more to that, but I love the idea, and if someone has an idea on how to solve this problem, let's talk about it. But
in the UK, even in the UK, where it's a much smaller country compared to the US, and we cannot communicate between hospitals, so I cannot look into a database of another hospital. It would be the same. You would need consent, data sharing. It's
it is still, we are not there yet. Ideally would be, once you're diagnosed, you can ask the patient, would you consent for your data? Yes, but it does, unfortunately, it's not, we are not there yet, but hopefully might happen in future. You need resources of the hospital to get this data entered, and it's not automatic, unfortunately.
One thing to add, related to that in a way, from my understanding, right, when family member got diagnosed originally, there was only one place in the United States that tested for Titin. Am I correct in saying that? But now getting that into the panels, right, where more and more physicians, so before to get tested, originally it was a full genome sequencing, right, and now it's this, and now if it's in a general panel, I think you're going to be picking up more people, and so if the physicians could just say, oh, I see a neuromuscular difficulty, I'm just going to send you for this genetic panel, you're going to get, right now you're seeing people in Titin popping up. All I see on Team Titin, more and more and more people being diagnosed, because I believe some of the work Sarah did in getting, getting that information and getting it a regular test to pick it up, and then it'll lead to, I think, being able to get more people in a database. Is it accurate, what I'm saying, or is it? Oh.
Yeah, and one of the cool things about some of the genetic testing companies that are getting more Titin data is they actually just were published on a cohort of over 74,000, and we learned a lot from that. Dr. Vata from Invitae published a recent paper on what did they learn from the sequencing data from 74,000 anonymized samples, and it was really informative.
That seems like a more achievable step, right? Like the data sharing is so big, so we can start with something that we can't.
If I can add one comment maybe on the ICD code, like on the one hand, it's kind of a blunt instrument, right? Because we're having one code for like skeletal and cardiac and all of this. But on the other hand, I do think it's helpful probably for that, maybe establishing numbers, because with the ICD-10 code, there's a lot of companies out there working on like real world data that like gather somehow anonymized patient data from most of the large health systems in the US. And with that ICD-10 code, potentially with a second criterion, like, diagnosed muscle weakness or cardiomyopathy, it's pretty easy to like go through the data once you have that code as like a starting point. And so I think that could really maybe help out, at least from a, establishing some numbers perspective.
Thank you. Well, we need to finish wrapping up because we need to head to the mental health sessions, which are starting next, and they're on a different floor, and there is some delays with the elevators. So, but it looks like, did you have one?
Well, I just wanted to say for the families,
Chris,
and for the folks that are this, sorry. The other complication with this, with identifying numbers and trying so hard to get Titin codes and to get clinicians and doctors to understand what Titin is having spent many, many years, more than a decade, trying to educate doctors. Like, oh, this is what this is. This is what Gigi has. Look, here, hold on. Let me like draw up the document and show it to you, or send you letters or whatever. Is that at the same time, for families, in order to live a good life and have care and have insurance, you need to know that if they don't know what Titin is, they're not going to approve it. So Gigi has muscular dystrophy. She doesn't have muscular dystrophy. Everybody here knows, like, that's like this umbrella that doesn't really capture it. It's actually congenital titinopathy. But for the purposes of insurance and doctors, I need to say muscular dystrophy or they don't understand. And I don't know that new families, especially ones with young children coming into Team Titin and other places too, understand that. They really need to know exactly what their kid has, but they also need to know that they're up against it when it comes to communicating and educating, and it's not our jobs to educate the entire medical profession. We have to make sure that we have that nomenclature, the words, to get the insurance coverage we need, to get the care we need, and to make sure we're actually talking to people, we're talking the same language with the doctors. So just a comment.
I just want to finish with a huge thank you to you guys, all of you, and the community for being here. So thank you, everyone.